Today we're going to talk about, pretty much it's an introduction to the kingdom Animalia. But determining if an organism is an animal or not are different characteristics, such as these are organisms that are heterotrophic by ingestion, different from fungi, which were heterotrophic also, but they release digestive enzymes out into the food. The food would be broken down outside of their body. In animals, by ingesting food, digestion occurs inside of the body of that organism. All animals are multicellular and all animals are eukaryotes. Another characteristic of animals is that they don't have like a cell wall. Instead, they're the only organisms that produce an extracellular matrix, which is pretty much a substance very rich in proteins and carbohydrates. Another characteristic that is unique to animals, however, it does not define an organism as an animal because not all of them have, is the presence of nervous and muscle tissues. Okay, so only animals have those. But I want to emphasize that not all animals have them. One thing about development of animals is that animals have certain structures that other organisms don't have. Like they form a blastula and a gastrula. The main part of the life cycle is the diploid stage. We all know the only haploid stage in animals will be the gametes. After sperm fertilizes the egg, the zygote that is formed undergoes a period of very rapid cell division called cleavage. At the end of cleavage, this embryo is going to look like this, like a hollow ball of cells. This is the whole embryo, and this is like cut. So you can see that there is a cavity in the middle. This stage here is called a blastula. Up to this point, there is no differentiation among these cells. Any of these cells would be able to form any part of the body of an animal. However, after the blastula is formed, there is a new process called gastrulation that the blastula undergoes to form an embryo that's called now a gastrula. And during this process of gastrulation, what happens is that cells from the outside of the embryo move to the inside, forming a cavity within that other cavity. At this point, during gastrulation, these cells now are not completely undifferentiated anymore. Okay, they're starting to get differentiated, forming what we call embryonic tissues or germ layers. And this little cavity within the cavity is the beginning of a digestive system. Still in terms of development, most animals are going to have something that we call the larval stage. And what a larva is pretty much an immature form of that animal that is morphologically different from the adult. And not only morphologically, but also in terms of needs. For example, think of a caterpillar and a butterfly. The caterpillar is completely different from the butterfly, not only in terms of morphology, but in terms of what he eats. Caterpillars usually eat leaves and plants, whereas butterflies are going to drink nectar. Butterflies fly, whereas caterpillars crawl. So they're very, very different ecologically and morphologically. Some animals that have this larval stage that's different, they're going to have to go through metamorphosis and then kind of become a juvenile, which is going to resemble better an adult. A juvenile then is an animal that resembles the adult, so they look kind of more similar, but it is not mature yet in terms of being able to reproduce. Here's an example of the development of a sea star. Another characteristic related to development in animals is that animals have a series of genes called Hox genes, and we talked about this when we talk about evolution. And these genes are important in the regulation of the development of the form of the body or the body plan. So there are several characteristics in the body plan of an animal that we look at in order to actually differentiate an animal from another. The body plan is pretty much like the morphology, right? The construction of that body. One of the first things that you look at when you look at an animal is symmetry. Some very, very primitive animals like sponges, not all sponges do, but some sponges lack symmetry. There is no way that you can cut the body of a sponge and get two symmetrical parts. And this kind of in order of evolutionary order. We see the first animals to evolve, some of them lacked symmetry, then others, next ones, kind of had a type of symmetry called radial symmetry, which is that when actually, if you look at the sea anemone, it has radial symmetry. And what it means is that from top to bottom, if you kind of cut the body of that animal, you can cut in any plane and you get, no matter what plane you use, you're going to get two symmetrical parts. So these animals are going to have a top and a bottom. The top will be where the mouth is, but they're not going to have like a front or and back or left or right. You see, it's all kind of looks the same all around. And then the most kind of complex animals show a type of symmetry called bilateral symmetry, which the name suggests. Bi means two lateral sides, so it's a type of symmetry that gives the animal two sides. In our case, a right and a left. So if you look at the body of this crayfish here, there's only one plane that you can cut it in order to get two symmetrical parts. Think about your body. Your right is symmetrical to your left, but your front is not symmetrical to your back. Okay, so really there is only one plane of symmetry in our bodies. We are bilaterally symmetrical. And this bilateral symmetry kind of now creates a difference all around the body of the animal. So it's not all the same as in the radial symmetry. It divides that body into dorsal, which would be your back, for example, and ventral, which would be the belly side. Right and left will also divide into anterior, where the mouth is pretty much, and posterior, where the anus is. Another characteristic of animals still related to development is those embryonic layers that we were talking about that are formed during gastrulation. During gastrulation, there are the formation of two or three germ layers. So if you look at an embryo, so the outermost layer is the ectoderm. It's called the ectoderm, and this is a layer that's going to form the exoskeleton of some animals. It's going to form the nervous system of the animals. Then the innermost germ layer is called the endoderm, and this is going to be lining organs. So it's the tissue that forms the lining of your lungs, the lining of the digestive systems, and in between the ectoderm and the endoderm there is a third layer in some animals called the mesoderm, and this is going to develop into the organs, the muscles, and so forth. Okay, so there are these three possible germ layers. Now, not all animals have all these three germ layers. Okay, so remember, like some animals, like sponges, they lack tissues completely. They don't have any of those. Others are going to have two of these germ layers, the ectoderm and the endoderm, and we call these animals diploblastic. So remember that, okay, because that's going to be important in separating animals. And then there are some animals that have all of those three germ layers. So in addition to the ectoderm and the endoderm, it also has the mesoderm, and these animals are called triploblastic animals. Another characteristic that we kind of use to identify animals is the presence or absence of a fluid-filled cavity called the coelom. Even though we use it to identify animals, it's not really an important characteristic in terms of separating them into evolutionary groups. So what a coelom is a cavity that is formed completely within the mesoderm. So if you look at this earthworm and you cut it, here you see in blue is the ectoderm, red is the mesoderm, and yellow is the endoderm. Notice that the coelom, the cavity that is forming, it's forming completely within the mesoderm. It's completely lined by the mesoderm. We call these animals coelomates. Now, there are other animals like nematodes, for example, roundworms, that they have a cavity. However, this cavity is not completely lined by the mesoderm. So if you look here, it's lined partially by the mesoderm and partially by the endoderm. So this is not a coelom. This is called a pseudocoelom, which means false coelom. So we call these animals pseudocoelomates. And then finally, there are some animals like flatworms that don't have any cavity at all. Okay, so we call these acoelomates. Another characteristic that we use to separate animals is, again, in embryonic development. And we separate them into protostome development and deuterostome development. Protostomes and stoma is the same as those, like the same root as for the leaves. It means mouth. If you remember in the lab, when we look at stomata, they look like a little mouth, right? Two lips. So stoma is mouth, and proto means first. So in the case of protostomes, the mouth is formed before the anus. And another characteristic that we find in them is that they have spiral cleavage. So during cleavage, the cells, so if you look here, this is a protostome embryo, the cells are not aligned. So the kind of plane of cleavage is kind of offset, between the different layers of the embryo. And deuterostomes, on the other hand, they have radial cleavage, which means like it's all kind of in the same plane of division. And the blastopore, which is the initial kind of pore into the gastrula of the embryo, instead of forming the mouth, it forms the anus. So the anus is formed first in deuterostomes, and the mouth is formed after that. So deutero means second. Okay, so this name, deuterostome, means that the mouth is formed second.